Hey guys, so today we're going to be talking about chapter 7. So this chapter covers bacterial metabolism. So how do they metabolize? How do they get their energy? What do they do to produce their energy? So fair warning, this chapter is um it's very concept heavy, so bear with me. I will also be posting an additional supplemental video that sort of explains it a little bit better than how I do, but it's also there just for your help. But I will definitely point out the stuff that I feel that are big concepts and that I feel that you should be able to translate onto the test. So let's get started. So chapter objectives, we're going to be dealing with the biochemical reaction as to like what do these microbes do, and then also with their metabolism, how does that actually interact with us? And then look at this cute little giraffe picking his nose. I thought it was cute. Okay, so energy of life. So energy is absolutely key to survival for any form of life. So how do living organisms assemble themselves with smaller molecules? So the universe is becoming more and more random due to something that's called entropy. So entropy is defined by everything leading to chaos. Um, that's pretty much as far as I will go for entropy. If you want, you can definitely take a whole class over it. But for this class, just know that entropy is defined by everything leading to chaos or becoming more random. So, organisms that live, they have to be able to assemble smaller molecules in order to survive. So, such as like water, H2O, they need to be able to assemble those molecules for survival. And they do that by spending energy. So, such as, so here are just some little side note examples. So like shigellosis, um, he's a very common illness. A lot of times it happens like in children because kids are absolutely dirty. Also, it will happen a lot of times in developing countries where water supply is often contaminated. So Shigella species, it's a gram-negative pathogen, and this pathogen is usually not really causes like severe infections. But what it is that there is a auger that specifically indicates how he metabolizes. So within that auger, there is an ingredient called lactose. And with that, a lot of species will ferment that sugar. So Shigella and like Salmonella, they actually don't ferment it, whereas E. coli does. So based on how they metabolize, we can actually use that to our advantage to identify certain bacteriums. So the fact that if we were to grow Shigella species onto this I can't even say it. I believe it's like I think it's pronounced hetonon auger. So if you were to grow him on that, you would see that he does not metabolize lactose. Like he doesn't use that as an energy source, whereas E. coli does. And a lot of times, if you use augers like that, there's some kind of like color change within their colonies, and I'll actually show you that later on in the lecture. So there are different ways to classify organisms based on how do they utilize their energy. So we kind of already went over this a little bit, but just a little recap. There's phototrophs, autotrophs, and heterotrophs, and chemotrophs. So phototrophs are the ones that actually absorb light energy and they use that. Autotrophs, these are organisms that produce organic materials. So they will make their own. Whereas chemotrophs, they need to actually take in their energy source their carbon energy source. And then heterotrophs, these are the ones that also, oh, I'm sorry. Correction. So chemotrophs, I'm sorry, guys. So chemotrophs are actually the ones that will make their energy through chemical reactions. I'm sorry. Whereas what I was saying earlier was heterotrophs. These are the ones that need to take in their carbon source. So we are heterotrophs ourselves. Phototrophs are an example of like plants. Autotrophs, they can are also plants. And then chemotrophs, I don't know any chemotrophs off the top of my head, but that's okay. And the next slide is a table that shows everything in one little spiel. So the big thing I want to point out from this table is specifically the chemotrophs. So chemotrophs can be broken down even further. So if you see something that's called an organotroph, these are guys that use organic materials, whereas a lithotroph, they use inorganic materials. So if you are going to be classified as an organotroph, then there are three main molecules or elements that you are going to be utilizing, and that is carbon, hydrogen, oxygen, nitrogen, and phosphorus. So if you are an organotroph, then you use organic molecules, which are the five that I pointed out. Whereas if you don't use any of those five, so if you use, let's say, sulfur, then you are considered a lithotroph. And then also if you're anaerobic or aerobic. So aerobic means that you use oxygen in your metabolism, whereas if you're anaerobic, you don't use oxygen during metabolism. So definitely keep that in mind, guys. And then also be able to identify what is a potential energy source for an organotroph and for a lithotroph. So again, those five organic molecules are carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, so let's move on. So in order to yield energy, there has to be some kind of reaction. So you start off with something and then you end with something. And then the direction of that reaction will determine what kind of free energy you're using. So I know there's an equation on this slide, guys, but I'm not going to expect you to calculate that anything from that equation. What I want you to get from this slide is the difference between an endothermic and an exothermic reaction. So I definitely want you to be able to recognize these curves on an exam. Also, I want you to be able to tell me what is the difference between an endothermic and an exothermic. So an endothermic, at the end of their reaction, it will be cold, whereas an exothermic, you are releasing heat. So an exothermic, you release heat, so you're hot, and then an endothermic, you absorb the heat. Okay? So definitely be able to identify between endothermic and exothermic. That's all I want you to get from this slide. So don't worry about having to calculate this delta G, okay? Don't even want you to worry about it. Okay. Also, another thing that can aid in reaction is something called enzymes. So enzymes specifically, they help lower something that's called the activation energy. So they make the reaction go quicker, and they also aid the reaction in not having to use so much energy. So this blue curve represents a reaction without an enzyme. And as you see, it takes more energy and it takes longer since you have to use so much energy. Whereas if you add an enzyme, you lower the activation, meaning that it gets to the point of metabolism a lot quicker, and then you produce the same amount of products though. So definitely on an exam, I want you to be able to explain what's the purpose of an enzyme. So the purpose of an enzyme is to lower activation energy. That's the main purpose. So meaning that enzymes, they lower the amount of energy needed in order to get to your final product that you desire. Okay, so now we're going to go into what actual molecule do we use for energy. And with that, I'm going to show you a short little clip. And it's from The Office, so hopefully I have somebody, at least one person in the class that is an Office fan. Here we go, guys.
Parkour! Michael! Parkour! Parkour! Extreme! Parkour! Parkour!
This is Parkour, internet sensation of 2004.
Okay, guys. So the reason why I showed you that clip is because personally, I think it's kind of funny. These guys are absolutely ridiculous. But another thing was because these guys are using a lot of energy. So they're using a lot of energy of a molecule called ATP. So ATP is specifically the molecule that we use in order to do metabolism. So there are two forms of metabolism. There's catabolism and anabolism. So catabolism is specifically the breakdown of a large molecule to a small molecule. So you are releasing energy, whereas anabolism is where you are building up something and that requires energy. So you have to utilize it. So definitely be able to define the difference between them. And then specifically, the molecule that we use also, is something called ATP, so adenosine triphosphate. So we use these to carry out energy. And then we also use another molecule. I'm not even going to attempt that name, but it is called NADH. So ATP is this molecule right here. Fabulous. So it's all this beautiful molecule. So, like I said, the reason why I showed you that is because those guys are using a lot of ATP whenever they're trying to do their ridiculous parkour. So, definitely know what two energy molecules do all organisms use. Every single organism uses ATP and NADH. Okay? So, whenever we are doing catabolism, this is what is called a redox reaction or an oxidative reduction reaction. These are the ones that reaction specifically involves the transfer of electrons between different molecules. So definitely remember something that is called oil rig. So it's oxidation is loss of electrons, meaning that they are getting rid of them, whereas reduction is gain of electrons, so they are gaining an electron to their molecule. So definitely know the difference between oxidation and reduction. That's pretty much as far as I'm going to go with that. I'm not going to expect you to actually know how to do this mechanism right here. I'm not going to expect you to know how to do that. It's not really relevant for y'all. But I do want you to know what is a redox reaction. So a redox reaction involves the transfer of electrons between molecules. And then with the NADH, he specifically carries 2 electrons, and he will be donating them, or he will be so if he donates them, he loses them, but then also throughout the reaction he can also gain them back during metabolism. So just know that. And then whenever he does lose them, so whenever he does donate them, he becomes a different molecule, and it's called NAD plus. Okay, so definitely just know that. I'm not going to expect you to know, like I said, these reactions right here. The only reaction I want you to know is that if NADH is reduced, like if he donates his electrons, so if he undergoes oxidation, he becomes NAD plus. That is as far as I want you to go. Okay, so now we're going to actually transition into the actual whole metabolism cycle, but we're going to break it down into chunks. So the first part of metabolism is something called glycolysis. So the big thing about glycolysis is that you start off with a glucose molecule, so a basic sugar, and then what you do is that you break that sugar apart into something that's called a pyruvate. So for most bacteriums, the main pathway is called an EMP. But if you say glycolysis, that's perfectly fine. So like I said, in glycolysis what you do is that you start off with one single glucose molecule, and then it's broken into two pyruvates. And we're going to go into that. I'm going to show you a diagram. And then there are two parts of glycolysis that I want you to know. So there's a part where you actually consume energy. So it takes energy to break down that glucose, and that takes 2 ATPs. But then at the end, you will eventually yield 2 ATPs as well as 2 NADHs. And I'm going to show you a very simplified explanation. So like I said, you start off with glucose, and then what happens is that there's a breakdown of that glucose into pyruvates or pyruvic acid, either one, and with that you yield NADH and ATP. Okay, so on an exam, I would want you to be able to fill in the process. So be able to tell me, what did I start with, what did I end with, and what were the two side products. Now, like I said, there's an intermediate step right here that is not shown on this slide. So on the next slide is actually what it shows. So you have the pyruvic acid, and then from that pyruvic acid, you actually yield 2 of these. So you do this twice, but this is, like I said, very simplified version. So you start off with that pyruvate. So we're here now, okay? So pyruvate, and then from that pyruvate, you're going to turn it into something that's called acetyl-CoA. But then in that process of the pyruvic acid going to acetyl-CoA, you pop off an NADH as well as a carbon dioxide. Okay? So that is a very, very simplified version of the basic step of glycolysis. But that's all I want you to know, since this is not a biochemistry course. Now, if we were in a biochemistry course, this is something that you will see. This is absolutely god awful. I will not expect you to know these structures for the exam, but I just wanted you to see it so you know that it is not that simple. It's actually a very, very detailed process. But from an exam, I would just want you to be able to tell me that you start with glucose. From that glucose, you get 2 pyruvic acids, as well as 2 ATPs and 2 NADHs. And then from that pyruvates, you will yield an acetyl-CoA. And then from that process, from pyruvic acid to acetyl-CoA, you produce a NADH and then a carbon dioxide. That's all I want you to know in terms of those two slides. Like I said, I was just showing you this slide to show you that it is not as simple as I was making it to be. So I'm not going to expect you to know these god-awful structures. Okay, so there are alternatives if a bacterium can't do glycolysis. There are 2 additional pathways. It's called the ED pathway or the PP pathway. So depending on what happens in this first step from the glucose, depending on if it will actually go through the entire glycolysis process, will determine what is specifically yielded. But you will almost always yield some number of ATP and then some number of NADHs for the most part. I'm not going to ask you these different pathways in the concept of like what happens. I just want you to know that different species use different pathways, and them using different pathways is determined by what is their specific need. That's really all I want you to know in terms of that. Okay, now that's glycolysis. Now, if they can't do glycolysis, then they do something that's called fermentation. So fermentation will occur whenever electrons of NADH are put back into the pyruvate. So it kind of just cycles back. And then what happens is that they form a organic waste product called lactic acid. And then also with fermentation, no oxygen is consumed. So that means that it's an anaerobic environment. And then with respiration, it's kind of similar, but not really. This is whenever oxygen is present. And then also what happens is that the NADH, whenever they donate their electrons, there is a they throughout the whole process, eventually those electrons will end up with oxygen. So oxygen is used as an electron acceptor in respiration, whereas in fermentation, oxygen is not an electron receptor. Acceptor, not receptor, acceptor. Um, that's really all I want you to know. I just want you to know the definition difference between fermentation and respiration. Okay. And then here is a very simplified version of it. So, like, they will do the first part of glycolysis, then what will determine whether or not if they go the respiration route or the fermentation route is essentially their environment and what is actually necessary for them. So just know that there's a lot of factors that play into which metabolic pathway will a bacterium choose to do. Okay, so now why is this relevant to us? So this is relevant to us because we can actually identify certain organisms based on how they metabolize. So there are two forms that I show on this slide. There's one that's called the phenol red test, and then there's the McElroy. I'm sorry, McElroy, the Mac auger plate. So we will actually use a Mac plate in our lab. I believe we do that. I want to say week 6 we do it. It's pretty cool. But we'll talk about the first one. So this first one is called the phenol red test. So this it allows for acid production to be observed, and for acid production to be observed, there's a color change that happens. So whether or not if you at the end of after you inoculate and everything, whether if you have a yellow or a red will determine what environment was produced. So whether or not if it was acidic environment or if it was a basic environment. So in order to remember which color goes with what pH level, I have a really bad mnemonic, guys, but I hope y'all like it because I think it's kind of funny. So you think of red. Red is bad. So bad base. So think of like Megan Trainor. I'm all about that base, no trouble. So that's what I think of. So bad base. So if you produce a red environment, then it's a basic environment. Whereas if you have a yellow, it's an acidic environment. And then also a lot of times, sometimes gas is produced. But I believe for us, I don't remember if we actually do the phenol red test, but if we do, we don't look at gas production. So that's one. Now the other one is a MAC plate. So this one specifically allows certain bacteriums to ferment because in this agar, lactose is present. So if a bacterium can actually ferment that specific sugar, it will turn the colonies a different color. And I believe with this one, if the colonies are red, they are able to ferment. So they're able to use that sugar during their metabolism process. So that is why metabolism for us is relevant, because it can actually help us identify a bacterium. Okay, so next. So another part of metabolism is the TAC cycle. So this is whenever all electrons, so transferring of electrons to the NADH is occurring, and this happens in the mitochondria. So bacteriums have mitochondria just like how we have mitochondria, but they use it a little differently. But the big picture is still the same. They use it for electron transport. So in order for them to enter the TAC cycle, the original glucose that they consumed has to be completely broken down to the pyruvate, And then, as well as completely, that pyruvate needs to be broken down into carbon dioxide and acetyl CoA. So, with the pyruvate being broken down, there are two hydrogens floating around, meaning that there are two additional electrons that can form with NADH. So, that NADH then transfers those two electrons into the electron transport system. And with that, there's a lot of protons. So there's a lot of positive charges present because of those hydrogens. So hydrogens are positive. And then with that, there are a plethora of steps that happen. I'm going to show you a very simplified version that I just want you to know in terms of E. coli in a few slides. But just know that after the series of reaction that occurs, ultimately what you yield is NADH's FADH2. So he's important, but I really want you to know the NADH and ATP. So in a few slides, I'm going to show you the big picture of the TAC cycle. So this is the nasty cycle that I was talking about, but I really just want you to know that in order for the TAC cycle to occur, there has to be the previous steps have to happen. If they don't happen, then the TAC cycle will not occur. So what happens ultimately in the TAC cycle is that acetyl CoA that came from the pyruvate is utilized and further broken down to yield the NADHs, ATP, and FADH2. That's pretty much it. Oh, one last thing, the importance of the TAC cycle. So it builds three main things. I'm really only concerned about two for this class, but the three main things that it does build is amino acids, fatty acids, as well as nucleobases. So I'm mainly concerned about the amino acids and the fatty acids. So that is why the TAC cycle is important, because the bacterium actually utilizes it in order to make proteins for them. Okay, so now the overall process of catabolism from the original breakdown is to reduce that bigger molecule into a smaller molecule in order to do respiration. So respiration is this entire process where they are metabolizing, and they will use electron acceptors called primarily oxygen. Some of them won't use oxygen, and if they don't use oxygen, then they do fermentation. But for the most part, respiration is what they do. And then with that, they do two things will happen. There's oxidative phosphorylation, and then there's the proton motor force. So the reason why respiration is so important to them is so they can actually survive. So in the oxidative phosphorylation, what will happen is that you have these electrons floating around, and then you also have oxygen. So oxygen will pick up these electrons, so he's known as the final electron acceptor. And then what he will do is that he will generate a proton gradient outside of the membrane. And then the fact that there's a gradient, and this proton gradient is hydrogens. So there's a lot of hydrogens floating around the cell inside and out, but the gradient is primarily outside. And then what happens is the fact that this gradient is being made, ATP is able to be powered, and then since the ATP synthase is being powered, ATP is being produced. And if ATP is being produced, then energy is being yielded. And then with that, you have the proton motor force. So this is all potential energy from those proton gradients. And then the reason why that's so important is that it aids in flagellar movement. So it aids in their travel, as well as the fact that they have all this potential energy surrounding them that allows them to uptake nutrients from their environment. So it's definitely important. And then this is kind of just like a little side note, but the overall equation that's happening for respiration is that they started with glucose, they also had water present in their environment, and then they had oxygen present in their environment. And then once they undergo their entire respiration process in the presence of oxygen, water and carbon dioxide is produced. That's the big picture. That's the overall equation of respiration. And that also goes for us too. It's not just for bacterium in this case, it's also for us, but we're talking about it in the context of bacteria. Now, with this, energy is also produced. Oh, excuse me. But definitely know that there's a difference of amount of ATP that would be produced whenever you compare respiration and fermentation. Respiration is definitely a lot more advantageous. It's definitely a lot more productive in producing more amounts of ATP molecules as compared to fermentation. Because fermentation is kind of, it's more of a temporary solution for them to metabolize. It's not a permanent solution for them. So they only do it for the time being, and then they don't want to do it the entire time because it does take a lot of energy for them to do that. And then also, the amount of ATP that's generated at the end of this process will vary, but for the most part, it's probably a good solid 30-ish ATP molecules. And the beauty about ATP is that it's renewable. You can always break it down and build it back up constantly. So that's why ATP is such a fabulous molecule. Okay, we are almost done, guys. A few more slides. Okay, so this is the big picture that I was talking about. So we start with glycolysis, we produce the pyruvates, we produce the acetyl-CoA, and then we go into the TAC cycle. And then from that, we can go into the electron transport system, and then with the electron transport system, we're able to pump out some ATPs. So this is the big picture, guys. This is really what I really want you to get from this lecture. If you can explain this big picture to me, we are good to go. So let me just go over it one last time. You're probably tired of hearing about it, but one last time, guys. So in glycolysis, we start with glucose and we make 2 pyruvates. And then from this process, we pop off 2 NADHs and then 2 ATPs. And then those 2 pyruvates will get changed into 2 acetyl-CoAs. And then from this process, we produce carbon dioxide as well as additional NADHs. These acetyl-CoAs then enter the TAC cycle, and we produce ATP and carbon dioxide again, as well as NADHs and some FADH2s. So all of these guys, so the NADHs on top of the FAD2 and hydrogen protons, they all travel to the ETS cycle. And then what will happen is that this proton gradient of hydrogens will occur. And with all these hydrogens out here outside the cell, ATP synthase is able to be generated. So these hydrogen are the energy source for ATP to be made, for ATP synthase to be making the ATPs. So then it will pump out some ATPs, and that is how we ultimately will get our energy. So that's the big picture of all, like, roughly the 20 slides before. Now, specifically in the context of E. coli, he does something very similar, but he has something a little different. Rather than having like ATP synthase and everything, he uses different proteins, And these are specifically, he uses two different kinds. He uses the NADH1 and then SIPO. So what happens for him in order to generate um, his energy, he has to use these two proteins. And what ultimately happens is that the NADH1, what he does is that he transfers electrons to a molecule called Q, and then this Q carries these hydrogens across the membrane and everything. And then what happens is that he will finally get to that second protein, that SIBO, and that SIBO will take the electrons from that Q molecule with hydrogens on it and then start pumping out those hydrogens, and with that he's able to make that water byproduct. So that's just, he specifically uses those two proteins. So I know that was like a little nitty gritty as to like what molecules are being used, but I really just want you to know the names of his proteins and know the fact that he uses um, two different enzymes rather than our enzymes. So just be able to tell me what are those two electron transport proteins. So for E. coli, it's NADH1 and then SIBO. Okay? So we talked about metabolism in the context of whenever oxygen is present, but now let's talk about whenever it's not present. So there are some bacteria that can do respiration without oxygen. So a lot of times, if they don't have oxygen present to get those electrons, they will use normally two other forms of molecules. So a lot of times it's nitrate or it's iron. And then organisms that don't like being around oxygen, they have to have something to accept that final electron in order to finish their metabolism. And a lot of times for that to happen is they will use different substances. So specifically, what will happen for like an example in like cystic fibrosis patients, what happens is that they produce a lot of sputum, and then our lungs is an anaerobic environment. So then the fact that it's an anaerobic environment, a lot of times they'll get an infection caused by Pseudomonas aeruginosa. And Pseudomonas specifically is, he prefers to be in an anaerobic environment. And with that, he doesn't have oxygen present to accept that final electron to produce his final energy product. So he uses nitrate instead. So over here, I do show other examples of species that like to live in anaerobic environments. But if I am going to ask you a question over it, I'm only going to ask you in terms of the cystic fibrosis patient. And then also, what are the alternative molecules that can be used as an electron acceptor? And the two examples that I talked about is nitrate and iron. Okay, so I'm going to skip this slide because it's god awful and I don't feel that it's relevant for y'all. Okay, so 2 more slides, guys. So how do these microbes, how do they use their energy in order to grow? So a lot of times whenever they need to get bigger and everything, the cells will communicate and they will connect their energy yield that they make from their metabolism, and they'll use that energy in order to build parts. So that's called biosynthesis or anabolism, or the opposite of catabolism. So I definitely just want you to know that in order for them to actually undergo biosynthesis, so such as like repair themselves or make a membrane or a capsule or anything of that nature, they have to have an energy yield. And then whenever they do have that, they undergo something called biosynthesis. So I really just want you to know that everything is interconnected, whether it be breakdown or it will be buildup. It's all connected, and the big thing is that there has to be some form of metabolism for them in order for it to occur. Okay, so I know this is a lot of information, but just one last thing for biosynthesis. There are several requirements for biosynthesis. Um, there are four main requirements. There has to be an assembly of a carbon skeleton, so meaning that the microbe has to have carbon present. Whether or not if they make it themselves or they consume it from others, carbon just has to be present. As well as they have to be able to fix nitrogen, sulfur, and phosphorus. So other than carbon, they need nitrogen, sulfur, and phosphorus present in order for them to grow. And then also, they have to be able to exert energy. So, meaning if they have to spend energy, they have to be able to make it. So, they have to have ATP and NADH present in order for energy to be made. And then, lastly, they need to be able to actually reduce that carbon skeleton, meaning that if they were to make certain things, they have to be able to derive it from something else. So, that was just a really quick synopsis of what is needed for them to make something. So that was a very brief. That's all I want you to know about it. So that will conclude our lecture. So we went over some how do we categorize the different energies of life. So based on how a organism will produce their energy source. We talked about enzymes, the difference between catabolism and anabolism. We talked about glycolysis, fermentation, respiration, and the TAC cycle, as well as the basics of anaerobic respiration and then the requirements for biosynthesis. So that was a very rough lecture, guys. I definitely want to reward you with a cute little video of pandas playing because they're adorable. Who doesn't like pandas playing? Okay, guys, so I hope watching pandas slide down hopefully made this lecture a little less painful. If y'all have any questions, please don't hesitate to ask. And then also, like I said, I will post an additional supplementary video about bacterial metabolism that maybe the way she explained it is a little bit better than how I did, which is perfectly fine. Um, and that will conclude our lecture, and I'll see y'all in class.